Ebola virus is named after the Ebola River, where it was first found. The first human outbreak was in Zaire, now the Democratic Republic of Congo, in 1976. Ebola is extremely infectious, but only moderately contagious. Ebola is infectious because a very small amount of the Ebola virus can cause illness. Some experiments in non-human primates suggest that a single virus can trigger an infection. However, Ebola is not extremely contagious. Ebola is not transmitted through the air like influenza. It needs contact with bodily fluids from a contaminated person or exposure with a contaminated object. As of April 20th, 2015, the current outbreak had killed 10,730 people in 8 countries, although all but 15 of those deaths occurred in 3 countries, Sierra Leone, Liberia, and Guinea. The entire Ebola virus particle, the viron, is 8 nanometers in diameter and 800 nanometers long. This is really small. Red blood cells are between 6,000 and 8,000 nanometers in diameter. In the center of the viron is the nucleocapsid. The nucleocapsid is the protein shell of the virus, which contains the RNA genome of the Ebola virus wrapped around various proteins. Surrounding the nucleocapsid is the outer viral envelope. It is derived from membranes of the host cell. It has 10 nanometer long glycoprotein spikes. These glycoprotein spikes are used to interact with other cells. They are critical for the survival of Ebola virus. The only part of the virus responsible for attachment, fusion, and entry of the target cells are the glycoprotein spikes. These spikes also account for most of the differences between different types of Ebola virus. Between the outer viral shell and the nucleocapsid are different proteins. Ebola chooses the specialized immune system cells to attack first to quickly overwhelm the body. It does this by infecting the dendritic cells first. These cells are the first cells to induce an immune system response. They act like the brains of the immune system. Ebola enters the cells by using its glycoprotein spikes to bind to the dendritic cell's receptors for cell transport through the plasma membrane. Once it gets inside the cell, Ebola dissolves its outer viral envelope and releases its genetic material. Once inside the cell, Ebola uses the cell to produce more viruses. Not only does the Ebola virus use the dendritic cell to manufacture more of itself, it also makes the dendritic cell tell other immune system cells to kill themselves. This leaves the body very unprepared for a viral infection. Once the dendritic cell is saturated, the Ebola virus dissolves the cell plasma membrane and releases millions of Ebola viruses. At this point, there are billions of viruses in the body. At the same time, Ebola viruses have infected the liver. They easily penetrate the liver and begin to cause organ failure and more internal bleeding. Ebola then kills the guard cells of the body, the monocytes and macrophages. But before it kills these cells, it makes them open up the blood vessels and release fluids into the body. This is usually good, but here it is very bad. Neutrophils are activated. They are not supposed to be fighting viruses, and they do a lot of damage to the body. They also tell the blood cells to release even more fluid. This causes the internal bleeding now synonymous with Ebola. Tough luck if you get here. There are currently no proven effective antiviral medicines available for Ebola. The care patients receive is all supportive to deal with the side effects of Ebola. All the supportive treatments that patients received are aimed at stopping organ failure and giving the body time to mount an effective response. Patients are provided with fluid to help with fluid loss through internal bleeding. Patients are also given oxygen. There are 3 types of vaccines in use that we will talk about: live, attenuated vaccines, inactivated vaccines, and subunit vaccines. All 3 of these vaccines work by giving the body a small part of the virus that the body can then save for use against later infections. Live attenuated vaccines are a less virulent strain of a virus. They could either be found in the wild or created in a lab. Inactivated vaccines are made by killing the virus. They sometimes need multiple doses. Subunit vaccines are made by only including a small part of the virus in a vaccine. A 4th type type of experimental vaccine. Recombinant vector vaccines are vaccines where the genes that code for the production of a specific antigen are injected into the body inside of a carrier virus. Once the vaccine enters the body, an antigen-presenting cell captures it. The cell breaks apart the vaccine and presents parts of it on the cell's surface. The antigen-producing cell then travels to a lymph node and activates a helper T cell. The T cell alerts nearby cells of the presence of an invader. B cells are also activated. They can either turn into plasma B cells or memory B cells. Plasma B cells are the body's antibody factories. They produce antibodies specific to the virus. Memory B cells are kept in the lymph node so the body can identify a virus or bacteria in the future and quickly produce more antibodies for it. The plasma B cells create many antibodies that are made specifically for the virus. These antibodies attach to the virus and prevent it from getting into a cell and also mark that cell for extermination by the immune system.